According to the cloud. I must say a little bit of a disclaimer here. This is complicated, can be complicated stuff. So I recommend you watch these videos, these lecture videos, as many times as necessary to really get this down. It will help. And don't forget, you must read your book. I know it's difficult and it feels like sometimes you don't understand, you're not absorbing any of it, but your brain is absorbing more than you think. And the more times you hear these stories and these words and these terms and all that stuff, the more it will tattoo it on your brain. So I'm going to hit my first whiteboard. Where we left off last time was just beginning to speak about the sliding filament model of contraction. So I'm going to start at the beginning. I already went over the first part of this story, but I'm going to repeat that first part rather quickly. So let me get a black pen here. I'm just going to write, whoops, that's no good. I want a pencil. I don't know why I have this weird font. Let me see if I can fix that. Yes, I can. There we go. It's supposed to say sliding filaments. There we go. Sliding filaments. It's a theory, but it's also what they call the model, M-O-D-E-L, of contraction. I just write these titles out so that you can sort of follow along in your book as to what section we're talking about. So the sliding filament model of contraction starts with, it's a nerve cell innervating a muscle cell, causing it to contract. So real quickly here, I'm going to draw a muscle cell. And I don't know if I'll have time to label everything, but I can certainly say it, and I'll try to write the key words. This is a nerve ending. It's called an axon terminal. So this stem of a nerve cell that this is just the very end of this long process. And this is called an axon, and the end of it's called an axon terminal. So that's just some language. When I say it, you'll know what I mean. So this is the neuron up here, N-E-U-R-O-N, and this is the actual muscle fiber. And you can see when a neuron, it's actually a motor neuron, when it stimulates a muscle cell, there's no physical contact. There's a space or a gap here. That's called the synaptic cleft. And I labeled that the last time. So step one is an electrical impulse travels down the axon to the terminal end, the axon terminal. And that electrical stimulus, I'm going to represent it as this goofy line here, sort of electric line coming down. It travels down the membrane of that neuron. The electrical stimulus is called an action potential. So when I say an action potential, I mean an electrical stimulus. So first, step one, an action potential travels down the axon and arrives at the terminal end of the neuron. And what that does when this electricity comes down here and gets to the end, it causes calcium ions, Ca++, to go into the cell, the neuron. So it's called influx. So it causes calcium influx. Inside of this neuron is stored chemical in these bubbles called vesicles. That chemical is called neurotransmitter. So I'm just going to label this neurotransmitter in a vesicle. That just means in a bubble, okay? When so electrical impulse comes down, calcium goes in. Calcium binds actually to this membrane right here, and it causes this thing to go to the end, this vesicle to migrate to the end and release its chemical. Now, in the case of a skeletal muscle contraction, that neurotransmitter has a specific name because it's a very specific chemical. It's called acetylcholine. Sorry, let me get my spelling down. It's late in the day. I've been teaching since 7:30 a.m. So acetylcholine, that's the name of the neurotransmitter. So back to the beginning, electrical impulse travels down. When it reaches when the action potential reaches the axon terminal, the end, calcium fluxes in, causes this vesicle with acetylcholine in it to migrate to the end, and the vesicle releases the acetylcholine into the synaptic cleft, this space. The acetylcholine diffuses across this space. That just means it moves from an area of higher concentration to lower concentration. And then that acetylcholine binds to receptors on the motor end plate. This indented area of the muscle cell right here is called the motor end plate. And there are these little rectangles I've drawn represent receptors specifically for acetylcholine. So it goes electrical, chemical, and now when the acetylcholine binds to its receptor here, it causes an electrical stimulus to travel along the sarcoplasm of the muscle cell. So we've got electrical, chemical, electrical again. Now, that electrical stimulus, it's also called an action potential. It travels on the outside, the plasma membrane of the muscle cell. It's called the sarcoplasm. Sorry, the sarcolemma. As it travels across the sarcolemma, it gets guided inward by an organelle called a T-tubule or transverse tubule. So I'm going to draw some of those. T-tubules or transverse tubules, as T-tubule, that's what it's short for. They are invaginations in the plasma membrane, in the sarcolemma of the skeletal muscle cell. All right? So the whole purpose of this T-tubule is just to guide the electrical impulse from the surface of the muscle fiber inward to the interior of the muscle cell. When that action potential is guided inward, at the base of these T-tubules is another organelle. I'll use green. That's sort of wrapped around the base of the T-tubules. And they're all connected to each other. These big boxes that I'm drawing are called terminal cisternae of the sarcoplasmic reticulum. Everything in green is the sarcoplasmic reticulum. It's just an organelle. And those bulging rectangles that I've drawn at the base of the T-tubules are part of the sarcoplasmic reticulum that stores a lot of calcium ions. All right. Remember when we were studying the skeletal system, we said how important calcium is in the body? Well, here's one great example why. We need calcium to release neurotransmitter, and we also store calcium in those green pouches, in those green sacs of the, I'll label this. This is called a terminal, should be in your glossary from the last lecture, cisternae with an E, it just means more than one, of, I'm just going to put the SR, that stands for sarcoplasmic reticulum. What's in there? Calcium. Calcium ions are stored. So I'm going to just use red so that you can see my writing. As this action potential travels down here, causes calcium to come in, causes acetylcholine to be released across the synaptic cleft. When that acetylcholine binds, causes another action potential on the surface of the muscle cell. That action potential is guided inward via a T-tubule. When that action potential, that electrical impulse reaches this area right here, that electricity, that action potential causes calcium ions to be released from these storage sacs, the terminal cisternae. So I'm going to draw that. Calcium ions get released and calcium floods the interior of the cell all over the place. All right. That's chapter 1 and 2 of this story, or you could call it 1, 2, and 3. So I'm going to leave this whiteboard up if I need to refer back to it. Now we're going to see what happens to this calcium that was released from these terminal cisternae of the sarcoplasmic reticulum. So I'm going to call up a new whiteboard. And now I'm going to magnify the muscle fiber cell even more. So the previous page, let's just say that's magnified 1,000 times or whatever, 1,000 or 5,000 times than what we can see with the naked eye. Now I'm going to magnify it a gazillion times. So it's super high magnification, and we're going to look even more microscopically. Deep down inside of the muscle cell, there are myofibrils, those long cylindrical organelles that contain those contractile filaments, myofilaments. So I'm going to draw a myosin, a thick filament. And I'm just going to draw one myosin head on it. Technically, there's a lot of them, a lot of myosin heads, but it's more simple, easier to understand and see if I just draw one. And this is going to be an actin molecule, the thin filament. So this is deep in our muscle cells, and it's really microscopic. Remember, actin is the thin filament, and myosin is the thick filament. Remember, this myosin molecule is attached to that vertical bar called the M line. All of these things are just basically polypeptide chains. They're pieces of proteins. Now, when a muscle is at rest, first let me say there are active sites on actin that the myosin head, that red golf club head, it can attach to these active sites, okay? And they're not all on the same side, by the way. When a muscle cell is at rest, there's actually another protein that covers these active sites on actin, covers them up so that myosin cannot bind. That grapevine-looking thing that covers the actin active sites is called tropomyosin. So tropomyosin is a peptide chain. Its job is to cover the active sites on the actin molecule when a muscle is at rest, so that this thing, the myosin head, cannot bind to actin. We don't want it to bind when the muscle's relaxed. But let's just say we want to contract a muscle. We want to lift something. You want to lift a notebook or a chair or whatever. Your muscles need to contract in order to do that. Well, the first part of that story we've already gone over. Your nerve cells send an impulse to the muscle cell. And after all that chain of reaction happens, calcium floods the inside of the cell. Now, this is what that calcium does. On tropomyosin, on this molecule, it's blocking actin active sites. There are little tiny 3 amino acid tabs, they're little tabs like this, and they're attached to tropomyosin. Okay. Those little green tabs are called troponin, Troponin. When calcium floods the inside of the cell from the first part of our story, this is what it does. Calcium ions bind to troponin. So troponin binds calcium ions. When that binding occurs, when calcium sticks to troponin like this, it causes the molecule to change shape and lift tropomyosin off of actin. And when tropomyosin gets lifted off of actin, we have an actin molecule that its active sites have been exposed. Okay. And here's the tropomyosin. It's been lifted off. Here's the troponin molecules. They have bound calcium. And in this process, tropomyosin gets lifted off of actin, exposing these active sites. Now the myosin head right here is free to attach to actin. And the whole reason that we want it to attach to actin is so it can bind to it and then pull it toward this bar, toward the M line here. So my drawing might get a little messy now, but I need to explain the process. So calcium binds to troponin. That lifts tropomyosin off of actin, exposing the active sites on actin. Now myosin is free to reach up, bind to actin, and pull that actin molecule toward the M line. If that happens, and literally what happens is myosin reaches up and grabs it, pulls it a little bit, and then it lets go, and it reaches up and grabs it again and pulls it further, and then lets go, and then it reaches up and grabs it again and pulls it further. It's like a ratcheting effect. When that happens, actin, here are actin molecules. Here are myosin molecules. There's the M line. When this reaches up and pulls actin toward the M line, here, it also happens on the other side toward the M line this way. That's one sarcomere. And then it happens down here again, this way and this way, always toward an M line. That's what causes the entire muscle to get shorter. It contracts, meaning it gets shorter, and it produces energy and force when it does that. It enables us to lift our bones, basically move our bones and anything that's in our hands or on our feet. So that is pretty much the whole story. When myosin lifts up, attaches to actin, and pulls it toward the M line, pulling of actin toward the M line has a name. That process is called the power stroke. So that term, the power stroke, literally refers to actin being pulled by myosin toward the M line. Okay. That's a really long story that's important for you to know backwards and forwards. If we were in person, that would be your essay question on the exam. I would say, okay, explain or describe the events at the neuromuscular junction and what ignites the sliding filament theory, and that would be telling me that story. Since we're not in person, you're not going to have to tell me that entire story from front to back, but I will ask a lot of questions about that whole process on the next quiz. Last but not least, there is one bit, an addendum to this, sort of an epilogue. This thing right here, the myosin head, we've explained how it attaches, sort of how it attaches to actin and how actin can be pulled, but this thing has to have energy in order to do the pulling. So the next whiteboard I'm calling up is going to describe how the myosin head obtains its energy to do the pulling, okay? So let's do that here. This is called, the book calls it the cross bridge cycle. The cross bridge just refers to myosin head attaching to actin. You think of it in parentheses as how the myosin head gets its energy. First and foremost, how myosin head gets its energy is this molecule right here, ATP, adenosine triphosphate. And I'm not going to draw the fancy version, the complicated version of ATP. Basically, it looks like this. There's 3 phosphate molecules. That's where the name triphosphate comes from. It's attached to a ribose sugar down here. There's a little oxygen there. So this is just a pentose sugar. And then There's an adenine molecule, so it gets its name, adenosine triphosphate. Sort of looks like this. That's the chemistry of it. I don't really care that much about the chemistry. What I want to explain to you is this is an ATP molecule. How this molecule stores energy is in these phosphates. When we break one of these phosphates off of ATP, it transfers energy to another molecule like this. So the first phosphate, it's called the terminal phosphate. When it breaks off of ATP, that's going to give the myosin head energy. So I'm going to show you what this cycle is. I'm going to draw a myosin head. That's the thick filament, myosin. And remember, actin is up here. I'm not going to go through the whole tropomyosin, troponin thing. Let's just say tropomyosin has been removed. The active sites have been exposed. I'll just draw a few of them on here. Those are the sites that myosin can attach to. Now, this is how myosin actually gets its energy. First, an ATP molecule attaches to the myosin head. That's step one. So I'm going to draw an ATP molecule. It's attached right here. Then the ATP molecule splits its terminal phosphate right here, breaks it off, and I should have drawn my myosin molecule a little bit differently, so let me do that quickly. Let's do, when ATP first attaches, the myosin head looks like this. It's more bent backwards. When that ATP molecule splits, so ATP splits the terminal phosphate, that just means the last one in that line. And so when it does that, it causes the myosin head to move. And it's like cocking a gun, that little thing on the back of a gun, not the trigger, but the thing that you pull back that cocks a gun. So it causes myosin head to, just to remind you of what it's doing, it changes position to cock. I'm going to put like a gun. So what it does is it causes the myosin head to do this, move from this position that I've drawn to this position, sort of an open position like this. Now the P and the ADP, when you break off that last phosphate, is called ADP because there's just two phosphates left on it. ADP plus P are still attached, but because this broke off, it caused that myosin head to go from this direction to this direction, all right? Then, I'm just gonna get my eraser out here to clean up my drawing. So, ADP and P are still attached to the myosin head, and the myosin head now attaches to actin. So I'm gonna continue my actin molecule over here. In this cocked position, now the myosin head reaches up, attaches to the actin site, and we get the power stroke. Because there's energy, myosin can pull that actin molecule toward the M line, called the power stroke. So the myosin head pulls actin toward the M line. The myosin head, now that it's attached up here, and it's, let's just say it's pulled it, so now it looks like this, it stays attached to actin until a new ATP molecule attaches to it. So it looks like this after it's pulled, and there's an ADP plus P still on it. Now we've got a new ATP molecule out here. It attaches to the myosin head. The myosin head, after it attaches, can release from actin. The whole myosin molecule releases from actin. Remember, it's in this pulled position. And the old ADP and P leave. And the new ATP molecule is now attached to it, and it can do the whole thing over again. So that's why it's called a cycle. As long as we have available ATP molecules, that myosin head can keep getting its energy. So if we need to sustain a contraction for a long time, we have to keep using ATP molecules to keep re-energizing that myosin head. Just so you know, if you've ever heard of this term, rigor mortis, not sure if this is in your book or not. So if you're watching this, here's a question that I'm most likely going to put on the next quiz. Rigor mortis is a condition after a person dies that their skeletal muscles contract and they don't relax. You've probably heard of it. Now you can, I'll explain to you how that happens, and hopefully you'll understand why it happens. So when a person dies, calcium ions are released and these active sites on actin are exposed. The myosin heads reach up, grab actin, pull, and so we get this contracted state of muscle, but there's no new ATP being generated in the body because the person is not breathing anymore and they're not able to metabolize glucose. Those are the two energy sources needed to make this molecule. So they can't make any new ATP and their muscles have contracted. But remember, only a new ATP attaching to this myosin head can release it from actin. And so there is no release for like up to 30 minutes. It's typical that people who die experience this muscle contraction, continued state for up to about 30 minutes. Then once all of the cells start to break down and tissues start to break down, then there is release just from degradation of tissue for the muscles to go back into a relaxed state. All right, so I'm just gonna show you these boards again really quickly. There's step 1 and probably step 2 of how a muscle begins to contract, starting with the motor neuron. And then we looked at sort of a step 3 and 4. What this calcium does, calcium ions do, is they bind to troponin, lifting tropomyosin off of actin. Now, these active sites on actin are exposed. Myosin can reach up and grab actin. And then steps 5 and 6 are really, how does myosin get the energy to pull? Right here, this molecule. And when it splits this last phosphate on it, it gives myosin energy, cocks the head. Myosin reaches up, grabs actin, pulls it toward the M line. That's called the power stroke. New whiteboard. How long does a muscle contract? The duration of contraction depends on 3 things. Duration of muscle contraction. Remember, this is skeletal muscle contractions. That really depends on 3 things. First, the duration of the stimulus from the neuron. So that's ingredient number 1, a neural stimulus. So that acetylcholine has to be released. Now, if the nerve stops firing, then the muscle can't continue its contraction. So the duration of stimulus from the neuron is factor number 1. Factor number 2 is the presence of free calcium in the sarcoplasm of the muscle cell. Presence of calcium ions. I'm just going to say in the sarcoplasm. So we need that calcium in order to bind to troponin to lift tropomyosin off of the active sites of actin. And finally, number 3, the availability of ATP. So without all of these 3 ingredients, a skeletal muscle cell will not contract. And once it's contracted, if any of these ingredients ceases to either be available or to exist, the muscle will relax and it can't continue its contraction. All right, let's get on with a little bit less complicated stuff. I'm going to define some terms for you that are important. I'm writing, these are types of muscle contractions. You've probably heard of some of these words before. Isotonic versus isometric muscle contractions. This word, isotonic, just means when a skeletal muscle contracts, it usually moves something. It's called moving a load. If the contraction is strong enough and produces enough force, then we get movement of some sort. An isotonic contraction is defined as that the muscle length changes when the force moves some sort of a load. So I'm going to just write that definition, or at least part of it. I'm going to write muscle length changes. That means during contraction and load movement. Isometric, the muscle length does not change even though force is produced. So there is no change in muscle length. I'm going to write even though force is produced. So this is length changes, muscle length, I forgot to write does not change. All right. That's the difference between these two words. Now, there are two different types of isotonic contractions. So let me write those. I'm just going to write lowercase a. Your book first does, I have a note here that your book first describes concentric isotonic contractions. This means that the muscle length shortens during contraction. And force is produced as well. So it shortens and performs some sort of work. The other type of isotonic contraction is called eccentric. And all this means is that the muscle fibers or the muscle lengthens during contraction and some sort of work is produced. So the muscle still generates force, but the muscle is actually lengthening, not shortening. The best example I can give you of this is if you hold your arm out straight. I don't have much room to draw this picture. If you hold your arm out straight, there's your biceps, there's your elbow, there's your forearm, and you put something in your arm, let's say like a weight. So I'm going to make a fist. There's the thumb, and we're holding some sort of a weight in our arms. A concentric muscular contraction, isotonic contraction, would be when we bend our arm. When we do this, the biceps brachii here is shortening. That's why it bulges, and we bend our arm. An eccentric contraction would be when we then lengthen our arm while still holding the weight. It's still hard and heavy to do that, and there's still some sort of work being done and tension being produced, but the muscle's actually lengthening instead of shortening. And finally, an isometric contraction is when the muscle length does not change at all, even though force is being produced. A classic example of an isometric contraction would be, let's just say, if you were to walk up to a brick wall in your house or any wall on the interior of your house, and you push against the wall, you obviously can't move that. Or you walk up to a car and you try to lift it off the ground. Obviously, we don't have the strength to do that. So what's happening is, in that situation, our muscles are not really changing in length because we're not moving anything. but there's still force being created in our muscles. There's still tension being created. So an isometric, I'm going to show you an easy way to test an isometric contraction would be if you take your hands together like this and you pull apart on your arms, you can feel your muscles are still creating tension, but they're not shortening or lengthening the muscles themselves. I'm going to describe, I don't know if I did this yet or not, the difference between a tendon and an aponeuroses. A tendon, these are just attachments. They're connective tissue attachments to muscles that attach them to bone. A tendon is just a cord or rope-like piece of connective tissue that connects a muscle to a bone. They're points of attachment. An aponeuroses is the same thing. It's just a flat, sheet-like connective tissue that also attaches muscle to bone or to another muscle. So when we have a belly-shaped muscle or a long cylindrical muscle like this, the points of attachment are typically tendons to some sort of a skeletal structure like a bone, okay? An aponeuroses, think flat muscles that lie over the top of your skull, like cranial muscles, they're all very thin and flat, or your abdominal muscles, your abs, that's a sheet-like muscle that looks like this. And they're flat and long, and these attachment points to the pubic bone and up here to the sternum and certain ribs, those flat sheet-like tendons are actually called an aponeuroses. When a muscle cell forms, these are just more important critical terms. A myoblast, remember way back when we studied tissues, blast just means immature cell. Myo means muscle. So this is an immature muscle cell that gives rise to multinucleate adult muscle fiber cells. So this is an immature skeletal muscle cell. And when muscles are first forming embryologically, these myoblasts, they merge together. And when they merge together, they each keep their nuclei. That's how an adult muscle cell becomes an adult skeletal muscle cell becomes multinucleate. So I'm just going to write when they merge. That gives a multinucleate muscle fiber. That's a mature muscle fiber. Not all of these myoblasts embryologically merge to become adult multinucleate muscle fibers. Some of them stay in sort of this fairly immature state, and they become something called myosatellite cells. So myoblasts can either become mature muscle fibers that are multinucleate by merging together, or they can stay separate. They do not merge, and they become myosatellite cells. These actually are repair cells that if a muscle gets damaged, these myosatellite cells help with repair of damaged muscle tissue. Just checking my time. I will go into muscle metabolism in one more lecture. You know what, I'm going to cover it now and get it finished out of the way, but just suffice it to say, I am going to do another lecture on this muscle chapter because it is so involved, and I'll try to get it posted this weekend. New topic here is muscle metabolism. just think of muscle metabolism as how a muscle uses an energy source like glucose and oxygen to make ATP. I'm going to write how muscle fibers make ATP, or how they get or obtain ATP. The processes of this go back to Bio 1. That's why Bio 1 is a prerequisite to take this course, is we talk a little, I kind of expect you to know a little bit about glycolysis and cellular respiration. I don't expect you to remember every single detail of it, but the language should at least be familiar to you. Muscles store very little ATP. So they store some, but not huge amounts. So when a muscle starts to contract, the amount that's already in a muscle stored basically just starts the process of contraction. Then that ATP gets used up. Then the muscle has to somehow obtain ATP through other mechanisms, other ways. That's what this conversation's about. So ATP, because there's only very little of it stored in muscles, it must be regenerated somehow so that the muscle can sustain a contraction. So ATP can be generated or regenerated in one of three ways. So let's just say our muscle has started to contract. We've used up what little stored ATP there is in there. Now the muscle has to either regenerate used ATP or find new sources for it. That's what this conversation's about. So ATP can be regenerated in three ways. I'm going to number those 1, 2, and 3 on the next page. The first way is by direct phosphorylation. What that means is once an ATP molecule gets used up, it becomes ADP. So ATP, if we split a terminal phosphate off of it, it becomes ADP plus P. We need to somehow use this to make new ATP again. That's what direct phosphorylation means. Unfortunately, we can't just glue this phosphate back on to create that. We have to find a phosphate from another molecule and stick it on that ADP. So direct phosphorylation occurs in the absence of oxygen. So I'm going to write no O2. This can occur even when we're holding our breath or there's no oxygen present in the cell. We're going to start with the ADP. That's the used ATP. It's already been used up. And what we want to create is a new ATP molecule. We use a different molecule called creatine phosphate. And you better believe, you can guess that there's the phosphate that we're going to use, okay? So what we do is this creatine phosphate, we steal phosphate from it, and it just becomes creatine. And we take this phosphate off of creatine phosphate and attach it to ADP to get ATP. There's an enzyme in the human body that makes this reaction possible. It speeds it up so that we can really make it work. And so I'm drawing an arrow to that reaction, and that enzyme is called creatine kinase enzyme. Kinase. So creatine phosphate in collaboration with creatine kinase allows us to convert ADP back to ATP. So that's direct phosphorylation. This process in the human body provides for about 15 to 20 seconds worth of contraction. I'll refer back to that in a second or in a minute because let me do this. I'm going to go back to my previous page. Remember, first a muscle starts always with stored ATP. but that's just like maybe a second or two worth of contraction, just gets the process started. So we've used up the stored ATP. Now we go to direct phosphorylation. That will give us about 15 to 20 seconds worth of contraction. What if we want to contract the muscle longer? For example, let's just say you're going for a run, you're going for a jog. You put your shoes on, you take your first few steps, that uses up stored ATP. Now you keep running down the street. After about 15 or 20 seconds, you've used up what you can make with direct phosphorylation. So in order to keep running, the body goes to a process called anaerobic respiration. Your book calls it the anaerobic pathway. Way back in Bio 1, that's the same thing as glycolysis. Glyco means sugar, lysis means to break. So we're just going to break a glucose molecule. And by breaking it, it's going to produce energy. So what glycolysis really stands for is the glucose that's in our bloodstream and gets into our cells can be broken down. So glucose breakdown from blood or glycogen stores occurs in a muscle so that we can sustain this contraction. When we break that glucose molecule, that gives energy. I'm going to describe that pathway really quickly. It's going to be abbreviated version. It's not going to be complicated. We start with this molecule glucose, which is just a 6-carbon sugar. And when we break it into 2 3-carbon molecules, the result is pyruvate molecules. That's the result. In the process of just breaking down glucose into pyruvate, we gain 2 ATP molecules. So remember, this occurs in this, no oxygen is necessary. It's anaerobic. It occurs in the absence of oxygen. So this is going to get us a little bit more further down the road, further down the street. This can make ATP to make a contraction last, about 30 to 45 seconds. These are just estimates. So about 30 to 45 seconds, I'm going to put worth of contraction. So think of this, an Olympic sprinter. He or she's in the starting block. Just to get out of the block, they use stored ATP. Then to get just maybe 10 or 15 seconds up the track, they use direct phosphorylation. Now they want to run further. Their body goes through the anaerobic pathway of glycolysis. And we explained how I didn't do the nitty gritty of this. If this is a cell, a muscle cell, and glucose is out here in the bloodstream, when glucose enters the cell, it gets broken down into pyruvate. We don't have any oxygen necessary in this cell. When that occurs, we get two ATPs. When we break that glucose into 2 3-carbon molecules. As a byproduct of this, we also get, sorry, my board moved on me. We also get lactic acid as a byproduct. Now, lactic acid can cause our muscles to burn. So if you've ever exercised really heavily, either weight training or jumping up and down, plyometrics, whatever, when your muscles start to burn, that is from lactic acid building up in your muscles. When we stop doing that activity, so if I were to tell you to stand up straight on your 2 feet, bend over, touch the floor, and then jump up and try to touch the ceiling and keep doing that repeatedly until you are exhausted, most of us would last for maybe 30 to 60 seconds, and we'd have to start breathing heavily. But we could last for about 30 to 45 seconds with just pretty much holding our breath. Our legs then start to burn profusely, So it forces us to stop. When we stop, that lactic acid goes to the liver very quickly through the bloodstream where it gets broken down. And then that burn goes away. Now let's just say we want to continue an activity for minutes to hours long. Think of a marathon runner. This is pathway number 3 of regeneration, and it is called cellular respiration. This occurs in the presence of oxygen. Oxygen is a necessary ingredient. That's why if you start running down the street holding your breath, after about 30 seconds, you have to start breathing, and you start breathing very heavily because the demand of work that's being put on your muscles is demanding this right here, oxygen intake. So cellular respiration occurs in the presence of O2. And I'm not going to go through all of the nitty-gritty of cellular respiration. That's good news for you. But I do expect you to know a little bit about the process, a tiny bit about the process, and more importantly, the products, the constituents that come from it. So this, whenever we do muscle activity, this accounts for about 95% of all skeletal muscle activity. You go for a long walk, you're breathing. When we walk, we breathe 24/7. We have to in order to survive. But also that oxygen is an energy source for our muscles to sustain a contraction for pretty much anywhere longer than about 45 seconds. Now, I'm going to go back to my cell right here. Glucose, when it first gets broken down into 3 carbons, we get pyruvate and lactic acid, and we get 2 ATP. That's not super efficient. That's not many ATP molecules. If then we start breathing oxygen, this pyruvate will go to the mitochondria in the cell, and we have lots of mitochondria in our muscle cells. So there's a mitochondria. The pyruvate enters the mitochondria. The oxygen enters the mitochondria, and together those two things, it's called cellular respiration in the electron transport chain. Anyway, they produce up to 32 ATP per glucose molecule. So you can see when we introduce this into the equation, we get a lot more ATP when we break down glucose. That's super efficient, but it takes a long time. This one up here is not super efficient. We don't need oxygen, but it's super fast, and it occurs super fast in the body. This one takes much longer time, but it produces way more ATP. So now think of the marathon runner. Marathon runner puts on his or her shoes. He or she starts to run. The first thing that marathon runner does is use stored ATP. So these, don't be confused by these numbers. I'm going back here. The first thing, really, don't be confused by number 1, 2, and 3. Stored ATP is used always first. So if I ask you on the quiz, what's the first place a muscle goes to get its ATP? Stored ATP. Even though we wrote 1, 2, and 3, these are regeneration 1, 2, and 3. This is after the storage is gone. The muscle first does direct phosphorylation, then the anaerobic pathway called glycolysis, and finally the aerobic cellular respiration method. And when we introduce oxygen, we get way more ATP per glucose, but it takes a long time compared to the other reactions. I'm just making sure that I covered all of this. I'm going to stop there. I will pick up. I promise I'll do another lecture for you to help you get through all of chapter 9. I'll try it. I'll post this one tonight, and tonight means Thursday night. And then by Friday or Saturday, I will get a third one up to conclude this chapter. So let me stop recording.